But how it can recognize that DNA is a genetic material, it is a question. So to solve this question, many scientists perform experiment which show that DNA is a genetic material. In 1928, a scientist called Friedrich Griffith Frederick Griffith first of all demonstrated that DNA is a genetic material. Frederick Griffith performed his experiment on a bacteria called Streptococcus pneumoniae. Streptococcus pneumoniae, it is also called, this bacteria is also called Diplococcus pneumoniae. It is also called Diplococcus pneumoniae. Frederick Mister performed his experiment on a bacteria Streptococcus pneumoniae in 1928. It is one of the experiments which prove that DNA is a genetic material. Frederick Mister take two strain of Diplococcus pneumoniae, The two strain is first strain type 3 S strain, and second strain is type 2 R strain. Frederick Griffith take two strain of Diplococcus pneumoniae. First strain is called type 3 and S strain, and second one is known as type 2 R strain. Here S stand for smooth and here R stand for rough. Then what is smooth and what is rough? S strain of Diplococcus pneumoniae is called smooth strain and R strain is called rough strain. In a smooth strain, what happened? This is a cell of Diplococcus pneumoniae. It is called smooth strain because it having a covering around it, and this cover of this bacteria is called capsule. It means that it is a capsulated bacteria. But in a type 2 R strain, the bacterial is made up of only in such a way that it does not having this capsulated covering around it. So what happened? This is called capsulated Diplococcus pneumoniae and this is called non-capsulated Diplococcus pneumoniae. It is called S strain, smooth strain, and it is called rough strain. This S strain of Diplococcus pneumoniae is virulent. And this strain of Diplococcus pneumoniae is non-virulent. virulent means it having a nature to develop a disease. Develop a disease, but non-virulent Diplococcus pneumoniae does not develop disease. These two strains are taken by Griffith and perform his experiment. This experiment is performed in 4 steps. Here I am draw this diagram. This is very important diagram to understand the concept of Griffith experiment. This is called rough strain and this is called smooth strain. Rough strain is virulent in nature and smooth strain is smooth strain is virulent in nature and rough strain is non-virulent in nature. It means that the smooth strain provided disease in the mice. In his experiment, in the experiment of Griffith, he taken all the two strain, rough strain and smooth strain, and injected it into the mice. Because it take mice, why it take mice? Because the anatomy and physiology of a mice are very similar to the human beings. That is, it is one of the experimental material which is used in the laboratory to testing different types of medicines and different types of result. So rat are taken here. What happened? The rough strain, when injected into the mice by injected into the mice, and because it is non-virulent in nature, so the result will be obtained here. The mice is lives. It means that might